for this opportunity to speak up about the recent data of CNS neurodegeneration and retinal neurodegeneration. In our portfolio, we have wonderful diagnostic tools. We can use diaphragm. We could follow morphostructural changes and also to see neurodegenerative diseases of the CNS and to compare them with neurodegenerative process in the retina. And the first data was quite breathtaking. And many questions here. Can we use this way? Can we use it? Some say it's possible, the others say no. And what about glaucoma? Maybe glaucoma is just the ophthalmic replacement of CNS disease. Let's move together there. So what is neurodegeneration in CNS? Here on the slide, we have very recent definition, which is currently used for this nosological group. This is a polymorphic group of acquired or congenital progressive diseases, and the common features is the death of particular group of neuronal cells and increasing dysfunction. Symptoms of this disease are designed by the area of damage. And what is neurodegeneration of the retina? Here we also see the definition. This is again polymorphic group of congenital or acquired diseases that is associated with the death of highly differentiated retinal cells. Plus retinal pigment epithelium is added, which is associated with the neuronal death and leads to the visual acuity dysfunction, and clinical manifestation is also guided by the level of involvement. Neurodegenerative diseases of CNS have multiple manifestations. This is Parkinson, this is lateral amyotrophic sclerosis, Pick disease, Huntington disease, and Alzheimer's disease. Retinal neurodegenerative diseases presented by the spectrum. This is pigment retinitis, STGH, this is Leber amaurosis, this is age macular degeneration, and glaucoma. And today we talk about glaucoma. Here on the slide, you see multiple publications, national and international, and due to that, we managed to compare morphostructural changes that happen in the retina in the patients with Alzheimer's disease and also with Parkinson, and compare them with particular changes which are ongoing in glaucoma patients. We also see that morphostructural changes in Alzheimer's disease of the internal compartments, specifically ganglion cells, axons, and the optic nerve. But the characteristic feature is the loss of melapsin ganglion cells, and this is how circadian rhythms are deteriorated and they are becoming more active at the nocturnal time. In Parkinson's disease, we see the same reactions, plus reduce the density of rods and the death of amacrine cells and deterioration of synaptic links with the neurons. In glaucoma, morphostructural changes are very well known. This is neuronal fibers and the loss of ganglia cells with the remodeling of the optic head. The main optic disc. The main markers here are deposits of the neurotoxic proteins, which are amyloid deposits. In Alzheimer's disease, they are in fact filling in all the layers of retina. We don't see it in Parkinson. In glaucoma, we see deposits of beta amyloid just in the ganglion retinal cells, GRC, and tau belong second protein of degeneration, tau protein, also could be seen in the layer of ganglia cell and optic nerve in Alzheimer's disease. And just a few of tau protein could be seen in the internal nuclear layer in glaucoma. Two other proteins of neurodegeneration, alpha and gamma synuclein, which are typical for the Parkinson disease, which is considered to be synucleinopathy, and just a few in Alzheimer's disease and a little in glaucoma. And last but not least, all three diseases are characterized by hematoretinal and hematoencephalic barrier, and in case of that, we see micro and macroglia activation. Here on the slide, this is one of the recent publications that really shows all specificities, similarities and differences in the three neurodegenerative diseases of central nervous system that compares with changes happening in glaucoma. And we clearly see here how strong could be the degenerative proteins depots in Alzheimer's disease, and how degeneration protein changes in two different types of the diseases of the central nervous system. And in fact, in glaucoma, there are a little bit of different things that happen mainly: thinning of the upper layer of retina and domination of microglia and macroglia activation. If we compare hypotheses of neurodegeneration development of CNS, we see that four main hypotheses are dominating here. This is amyloid and tau hypothesis, where neurodegeneration is started. Deposits and proteins of neurodegeneration neurotransmitters, where the trigger mechanism is the neurotransmitter disorder at the level of glutamate, choline, dopamine, and serotonin, with the further reaching neurodegeneration and adding of other mechanisms. And last one is a calcium dysregulation that can cause specifically the launch of neurodegenerative processes. Among the hypotheses of glaucoma, there are two dominating, which is mechanical theory and vascular theory, that we all know very well and understand. There are many mechanisms of neurodegeneration in glaucoma, and they go in synergy, fostering each other, or could be reflected independently. And first of all, this is neurotoxic mechanism due to glutamate, nitrogen oxide, and calcium ion reflux, oxidative stress, and mitochondrial dysfunction, autophagia, and definitely different canonic and non-canonic pathways of inflammation. But today we will shift the option of studies towards activation of macro and microglia and intracellular interaction glia and neurons. So what is glia? Glial cells are known from the 1930s. They morphologically different from neurons. They cannot generate the potential of effect. They don't have chemical synapses. They can divide, unlike neurons, and their number is much higher versus neurons, and they have very wide spectrum of functions. We have to say that the spectrum of CNS glial cells is different from retinal glia. Specifically, in retina, we don't have endemocytes, but on the other hand, we see unique cell units in the retina, which is called Muller cell, which is absent in the brain tissue. If we look at where, in fact, these cell components are located, so let me explain you that microglia cells are normally located at the internal and external plexiform layer, and astrocytes localized in the neuronal filaments and ganglia cell of the retina, and majority of them is in the optic nerve, and when we see them in the myelinized axons, there we see oligodendrocytes attaching them that provide myelinization of the axons of the optic nerve. Besides, there is a difference in between glial cells into two types, which is micro and macroglia. Microglia has normally mesodermal origin, and if hematophthalmic barrier is ruined, here we can see microglia cells coming from the bone marrow structures. In fact, we can identify two populations, although this is more heterogeneous population, which is M1, which is constructive, and that is stable and provides multiple functions in the retina, and M2 that has macrophage-like functions and moving towards initial neurodegeneration. Speaking about macroglia, it comes from the crest, and these are Muller cell and astrocytes. And as I said before, oligodendrocytes could be attached to that. Microglia is homological to monocytes and macrophages, so expresses on its surface very typical features. Muller cell has unique spectrum of enzymes, for example, glutamine synthetase, that allows it to provide control of neurotransmitter system, glutamate glutamine, and the second protein, which identifies the uniqueness of the cell. So this is retinol acid, which is actively involved into the visual cascade. Three further proteins S100 beta, vimentin, and GFAP, which identifies how close mother cell to neuronal cell is. Astrocytes strongly express acid fibrillar protein, GFAP. Here on the busy slide, you can see the networking of astrocytes and ganglia cell of retina, and they're providing baseline functions. Microglia functions are multiple, so this is phagocytosis because of apoptotic bodies, also remodeling of the extracellular matrix due to the production of the matrix metalloproteinases provided by the regulation of synaptogenesis in the embryonal period. So for years, microglia cells were considered to be immunocompetent cells in the retina, and they are supporting and controlling immunoprivilege of this compartment. Also, it has controlling and regulatory function in the neuronal apoptosis, and they are participating in hematoophthalmic and hematoencephaly barrier, BBB. And by the way, they have very strong control and the regulatory function of the molecular hemostasis. Neurotrophic function is shared with macroglia cell. They are mainly secretory, protectory functions, as well as supporting of continuity, molecular continuity specifically, and they participate in the formation of myelin. Here on the slide, you can see molecular, roughly speaking, molecular anatomy of microglia, or you may call it the vocabulary. Using that, microglia cells are communicating in between. In glaucoma, we have very important cytokines and chemokines as TNF-alpha, IL-1, IL-6, IL-8, that are supporting pro-inflammatory signals. So on the left, you see the columns, and they are molecular classes. Here we see so-called super-sensitive receptors, transmembranic receptors, that as very sensitive radars could scan the extracellular cells, identifying the defects and then providing intracellular signal to react. So here you see toll-like receptors that help to identify genes, alien genes, and also receptors identifying molecular disbalance in the environment, and cytokine receptors, which allow to participate in the immune transmission and neuronal signal transmission from microglia. Here we see complement receptors. Complement is a system of the congenital immunity that participates in the production, so that means congenital immunity that's protecting neurons from the damage. It's difficult to overestimate the Muller function, which is a radial cell, and it goes from internal to the external borderline membrane without reaching the latter, and then interrupting its structure at the level of the joints. internal and external receptor joints, and it is supporting very stringent control of regulation in the retina due to its unique aquaporin pores presented on the side surface. The last publications show the supportive function of the density of the deep and the superficial plexus, and the most important function that Muller cells have. This is prevention of neurotoxicity because of glutamate clearance control. So all that is happening in the normal situation, but any moment it can be changed. So these cells from the partners, from the friends, could easily become foes. And for example, Muller cell could design excess of glutamate, and then we see the loss of synapses due to the cytotoxic death. So I would like to speak about the most recent data where it was proved that Muller cells have unique light conducting capacities. Like fibers, they take away the scattering and allow to create much clearer picture in the retina. Biologists even calculated particular indexes confirming this hypothesis. So the same glial cells can design and transmit these different molecules of interacting with microglia cells, and then to send the signals to neurons, and then they could provide very narrow specialized function. But in activation, these cells design lots of molecules that could provide a launch of neurodegeneration. And here they are transforming into the force. Well, very similar behavior we see in the microglia cell and Muller cell. There is some accumulation of such effects, and they could provide some neurotoxic functions. And we have already discussed that. Triggers of microglia activation are lipopolysaccharides, so that could be disbalance of pro and anti-inflammatory cytokines, components of complement degeneration proteins, and also active reactive oxygen species and nitrogen species. So in this situation, we understand that the process is associated with the reduction of the protection, and at the same time oxidative stress is accumulated with active oxygen species, which are superoxide anion, peroxide oxygen, and these molecules could provide direct and also mediated effect, causing the death of membrane structures of the cell. Not less important in the neurodegeneration is to see mitochondrial disorders. We have to say that mitochondria is not only the accumulator of the energy, it provides the control of 12 enzymes of the respiratory tissue, and in polymorphisms of DNA, we have to say that mitochondrial DNA is more perceptive to different disorders versus nuclear DNA. So this is how we see the accumulation of very different pathological molecules, whereas we see the attachment of reactive oxygen species, hydrogen peroxide, that launches the inflammation, that finally launches neurodegeneration. Mitochondrial disorders are closely related to calcium. Calcium in the extracellular space is in 100% higher versus inside the cell. So any reflux in the intracellular space would be associated with the molecular disorder that, with oxidative stress, could lead to the death of the membrane structures of the cell. And this is how we can get messages to launch the neurodegeneration. So the cell becomes activated. It changes its morphology. It obtains a marble-like structure and secretes lots of proteins, and these signal molecules start neurodegenerative processes. Where do we see the primary activation of glia? With the ophthalmic hypertension model of glaucoma shows that this process starts from the optic head at the level of the lamina cribrosa, but in the mice there is no lamina cribrosa, so these are glia cells. So there is neurodegeneration proximally and distally and starts the process of neurodegeneration. And here we see what happens at the morphostructural way. So here we see the same acid glycofibrillary protein loses its cell structure, discharged, and then neurons, axons of the neurons become very edemic, and they feel bad. And this is how it has been proved in experimental works that the earlier place of glia activation is cribrosa, and we see very rare glia activation in the preliminary layer or in the fiber layer. Inflammation is a very strong promoter of any pathological process, and neurodegeneration is not an exception from the rule. It has very different patterns, and we need to say that very different compartments react very differently to different mechanisms, pathogenic mechanisms. Specifically, a soma ganglia cell easily go into apoptosis with the death ligand activation. Also, for the apoptosis initiation, BCL2 proteins are important, and the most important is BAX. And if BAX is reduced, then protection of ganglial retinal cell and their survival is getting much stronger. Synapses. There is poor effect of different processes of complement protein activation. Also, dendrites and axons are poorly influenced by the cocktail of pro-inflammatory proteins. And this is how last publications emphasize that in this situation, to the different compartments in ganglia cells are influenced very differently. And what should be the consequence of that is very difficult to understand. It all depends on the individual conditions that ophthalmic situation develops. And this scheme demonstrates that all these canonic and non-canonic pathways of inflammation and complement are overlapping one in one specific point, which is a nuclear factor, NF-kappa beta. So, and then this vicious circle could be ruptured when we influence these tools. So for years, there was quite active discussion how autoimmune mechanisms could influence. Is it an epiphenomenon or is it an independent mechanism? So here we see multiple experimental studies saying that antigen-antibody reaction that creates this construction could be seen, which was proved on non-ophthalmic hypertension models, and morphological confirmations of the presence of this reaction of antibody dependent, which is activation at the level of adaptive immunity. Starting from 2013 up to 2019, we carried out of the series of immunological studies, and this is our small research group. And the data we obtained actually was the basis of 3 PhD theses. The studies were supported by Russian Foundation on Fundamental Studies. They were carried out in different sites. So that was Institution of Bioorganic Chemistry and Russian State Medical University and University of People's Friendship. So we extended the topography of the immunological reaction, and we got facts confirming that antigen-antibody reaction happen in trabecula, and they influence the changes of the trabecular network. And the similar confirmations of the immunological reactions were obtained at the level of the myelinated axons of the optic nerve, except traditional compartments at the level of ganglia cells. And also we got the confirmation that we observe immunological reactions at the level of bispigment or epithelium of ciliary fibers. So here we enjoy the presentation on the autoimmune inflammation. So that means proteome exchanges, the amount and the spectrum of proteins is changing, and they could really cause specific autoimmune reactions that we mentioned before. We have to say that the paradigm has changed in immunology. We used to believe that any excessive content of antibodies provides cytodestructive effect, but in the recent years, specifically on the ganglia cell of the retina, we see the confirmation that there are specific types of antibodies that provide protective function, and if they're reduced, it leads to the fact that the immune protection of the cell is reduced. Mainly, it refers to such unique antibodies like enolase and viron-specific enolase, which controls metabolism and utilization of the glucose. We know how important for the neurons metabolism in the system. So we obtained this data, and correspondingly we presented the data. They were presented by our international colleagues, and actually we got the same data from them with the particular increase of antibodies, and they got the reduction, they got the drop down, and they confirmed it morphologically. So we moved the next step, and together with serological mapping, we carried out morphological one. We found out where these antigens could be localized, and we compared the data with morphometric parameters of macula, and we got very good correlation showing the true link in between the process of morphostructural changes in glaucoma and the deficiencies of antibodies. And very similar data was obtained, similar correlations were obtained for the optic nerve heads and retinal fibers. The publications that are confirming our conclusions that the protection at the level of retinal ganglia cells is suffering in case of some strategic antibodies reducing or losing completely. It is offered to use such antibodies as very useful biomarkers for the diagnosis of severity progression. And our recent publications are showing the importance to study the functioning of particular case spaces, which are providing the launch of apoptosis and, in fact, regulate or identify whether the ganglia cell should live or die. Should live or die. And here you can appreciate the closed loop system where all these pathogenic mechanisms are in the same vicious circle. So we can rupture it via very different medical tools focused on the recovery of the mitochondrial function and immunomodulation. Thank you very much for your attention.